Since 90 years, politics and social scientists talk about the need to concile the global and local dimension. Global and local dimension about environmental issues, but about social cohesion and other policies. So thank you very much, Ailburu, to remember us the importance of this coherence between the big approach, big problems, and the local implementation public policies. Thank you very much. So we close the open ceremony at this stage. We thank all speakers, the words that provide a great context, and I think a very good framework with very different keywords for the coming discussions. The opening will be followed by 3 high-level panels today. The first panel is about climate change and biodiversity, both sides, climate change and biodiversity, and will point out the importance of informing climate change and biodiversity policies with best data and by integrating knowledge. The panel will be moderated by Maria José Sanz, Director of BC3, and we will be able to listen before Mr. Mark Wilkinson. Mr. Wilkinson was lead author of the primary fair data principles paper and lead author of the first paper describing a complete implementation of those principles over legacy data. He's a founding member of the FAIR Metrics Working Group. He studied at the University of British Columbia and spent 4 years at the Max Planck Institute for Zuchtungsforschung, I think so, Zuchtungsforschung in Köln, in Germany, pursuing studies in a mix of plant molecular and development biology and bioinformatics. He then did a research associate at the Plant Biotechnology Institute of the National Research Council in Canada, focusing on the problem of biological data representation and integration for the purposes of automated data mining. Now, he's developing a more biologically focused research is to achieve the introduction of precision farming into areas with limited resources and/or access to contemporary sequencing of informatic technologies. Mr. Wilkinson, the floor is yours.
Thank you very much. So I have to start by thanking the organizers for the invitation. It's very kind, and I was not expecting this. And I'm really pleased to be here with this audience. And thank you for the organizers also for making it completely effortlessly for me to get here. The logistics was fantastic, so thank you all. So I'm going to talk, I've been asked to talk primarily about FAIR, but at the end I'm going to segue into a bit more of the topic of the panel. So I'm going to talk about strategies and technologies for early wins. The talk is going to be not philosophical and not technical, but rather pragmatic. So to frame it, FAIR is intended to help machines help people. It should be replacing the time spent in data discovery and manipulation with time spent on thoughtful exploration of global knowledge. And I'm going to show you that it works. So it's not just a dream, and it's not just a bunch of nice words, it actually works. And we've also, however, experienced some notable failures, and if I tell you about those, hopefully you won't go down those paths. So, let's get started. There's the paper, right? And this was unexpected in my career, I have to say, a very significant event. It now has more than 13,500 citations. And I think that for me, this was the most important sentence in the paper. This necessitates machines to be capable of autonomously and appropriately acting when faced with a wide range of types, formats, and access mechanisms and protocols that will be encountered during their self-guided exploration of the global data ecosystem. And when I wrote that paragraph, I was not talking about people. I was talking about machines. So automated exploration and discovery agents. And there's some consequences from that. First, FAIR is, first, a mechanism to guide automated agents to discovery of task-relevant data. And as a consequence, FAIR is above all else about metadata. The second consequence is that if we want to create a web of data that can be appropriately, and there's a lot packed into that word that I might talk about later if I have time, appropriately reused by machines. It necessitates specific data publishing behaviors by the providers of the data. Those behaviors can be concretely described because they have to be coded, and of course we then code the software that leverages those behaviors, and we end up getting the FAIR repercussions. But the consequence of that is that fairness is, by definition, measurable. Right? So you will be measured on your fairness by automated agents. So I want to talk about these measurements because these are probably the most controversial part of FAIR at the moment. So the FAIR Evaluator was the first fully automated agent for testing the fairness of a resource. So I wrote this now 6, 7 years ago. This resulted from early pressures shortly, the year after 2016 when the paper came out. We were getting pressures from journal editors and funding agencies who wished to require fairness but didn't know how to check. Right? And so this is what the tool looks like. There's the address if you want to visit it. I think I can put up a... No, I can't. Okay. So this is what the tool looks like. What you do is you or any stakeholder provides the URL for any digital object, and this tool will run about 22 different tests measuring different aspects of fairness, and it will run those automatically, and it will give you an output. What's happening in the background is that there is a fairly a fairly complicated workflow I'm not going to describe because I'm not going to be technical. But there's a fairly complicated workflow for finding the metadata that is associated with the digital object. And this turns out to be extremely hard. Right? It's highly variable from provider to provider, and not everyone follows standards. So when I say very forgiving, it means that I'm very forgiving. Right? If you try, I will give you credit. And so the tests then work on that bunch of metadata. And so, for example, FAIR Principle F3, metadata explicitly include the identifier of the data it describes. What I do is I take the harvested metadata and look for any facet that seems to be a reference to a data record. And there's at least 18 possibilities that I search for that I find acceptable. Right? That's very generous and very forgiving. Right? So the evaluator has been used about 10,000 times on the public version. Full disclosure, I have a private version that my company runs, which has been run several thousand times also. And individual tests are also executed outside of the evaluator itself, so the individual tests are publicly available also. But they do occur frequently, but I don't measure the usage. Right? But here's the problem. So fair assessment is now a cottage industry. Everybody thought that it was really important to assess fairness, and everybody all at the same time started to build tools. So we now have 22 different independently derived fair assessment platforms. Most of them are based on questionnaires. A couple of them, including mine, are automated. All of them focus on metadata for the most part, but there's a problem. When you use multiple tools, you find that you can't compare the scores. How bad can it be? So this is a comparison of my evaluator. This is the commercial version, by the way, just again, full disclosure, with another automated tool called Fuji coming out of the Fair Impact project on the same digital object, which is a record that I created for the Duchenne Parent Project patient registry. This is mine, 20 out of 22 tests pass. This is Fuji, 2 out of 24 tests pass. That's how divergent the testing can be, right? And that's scary, because it means that we're not interpreting fairness the same way. And the FAIR principles don't help because they didn't tell us how to interpret it. And so the EOSC said, okay, we can't require FAIR for the European Open Science Cloud if we don't know how to measure it. So they called for an investigation that led to the creation of a task force on FAIR metrics and data quality, and I was co-chair of that together with, at the beginning, Carlo, and then Chris Schubert came in later on, established 2021. It's now completed its task. Another task force has just been started. We don't know who the co-chairs are going to be, but I am involved in that. The three key outputs, I'm not going to go into details, but the three key outputs with respect to FAIR testing were some framework for doing an apples-to-apples comparison between the different testing frameworks, a realization that we cannot do testing without some form of governance, and so we're starting now to look at governance models for FAIR testing. And if you don't read anything else, please read this, and all of the links are in the slides. The slide deck, by the way, is CC BY, linked, you can reuse it. This is an attempt to stop the chaos and to get people to adopt adopt a uniform and better metadata publishing paradigm called FAIR signposting. So if you don't read any of them, read that one. There's a project that has just started in January 2024 called OS Trails. This is an EOSC infrastructure project. Within the deliverables of the OS Trails project is a deliverable to harmonize FAIR assessment tools, workflows, and APIs, and also bootstrapping this global governance process around FAIR testing. And these activities are open. If you want to participate, contact me. Okay, now I want to show you some success. Now that I've shown you failure, I want to show you some success. So I've been involved in this project called the European Joint Programme on Rare Disease. And so let me describe to you what we did in this project, because it's extraordinarily successful in my opinion. Let's look at the scale of the problem. So the EJPRD in numbers, 1,800 participants, 35 countries, all 24 of the rare disease reference networks. This budget is much higher now than it is advertised here, because we've had additional funding coming in. 91 beneficiaries. This is the important number, 91 beneficiaries, 52 linked parties, and so on. 85% of the European rare disease community is there, and our challenge was, make that fair. Okay, how? That's a big, big network, right? 91 participants, not all of them have their own data, but many of them do. So, step one. And you're going to see this over and over again. Step one, focus on metadata. The first thing we have to answer is, what kind of data does each partner have? Right? And we have to answer that for a machine. So we created this metadata schema. It's based on the data catalog standard. We attempt to follow the European data catalog application profile. Every participating site publishes this via a semi-automated process I'll show you in a moment, but it's bootstrapped using Excel spreadsheets, which everyone is familiar with. The metadata publication platform we're using is called the FAIR Data Point. So this is an open source code base distributed via Docker images. It installs within a couple of seconds. It publishes metadata for humans, but it also, most importantly, publishes metadata for machines. It provides an easy-to-use editing tool, so end users can edit these DCAT records through web pages, and it automatically registers your existence in a central location, your existence, right? That central location then shows us who is a member of this federated network of FAIR data points. And so here you see, for the European Joint Programme on Rare Diseases, this is our index, and you'll see all of the partners are here. And there's an indication of their status. So these are the active ones. People can see, if they check themselves, they can see, oh, I've fallen out of activity, something went wrong. They can bring their FAIR data point back up. So this is a central registry of partners, and this makes it now possible for a computational agent to go to that registry and say, okay, who are the partners? And then individually visit them and explore their metadata and figure out which ones potentially contain data that's of interest to my question. This is all happening at metadata layer, right? So far we haven't talked about FAIR data. Metadata, metadata, metadata. Right? So am I suggesting that there's no point in making data fair, or making fair data? No, I'm not. But I'm saying that there's little point in making fair data until you get the discovery metadata right. And then you can start to get some benefit from the fair data, or more benefit from fair data. And we have to recognize that some data will never be fair. Some of it is highly sensitive, some of it is just too voluminous, and there's not much point in changing its structure in order to make it fair. So metadata is really the point of fair, not data. But EJPRD also did create fair data, and I'll show you briefly the end-user-friendly and reusable verification pipeline that we implemented and is now open source. So let me just describe the challenge again. So we need to make all of the data in this network work together. So there's about 50 resources that actually have data. Now they have similar data. So they're either patient registries or biobanks, but they have different starting formats. Generally, they're forbidden from sharing or moving their data. You heard in the opening talks this issue of data privacy and security and access. So as a consequence, the participants are going to have to do the fair transformation themselves, on site, and sometimes without even a fair expert seeing the data. Right, now that's a challenge, because these are not fair experts. These are often not even technical experts. These are registry owners, clinical-oriented people who don't necessarily know a lot about fair. So, how do we do that? Well, the way that we approached it is we started by building a fair data model that could represent more or less all of the different data that was collected by these registries. We called this the Clinical and Registry Entry Semantic Model. This is a snapshot of the core, the highest level. It's based on the semantic science integrated ontology from Dr. Michel de Bontier, who's at University of Maastricht. But it was finished by Pablo Alarcón, who's a PhD student in my group. So we share that model. Everybody gets a copy of it, but they don't need to understand it, because all they do is they generate CSV, comma separated value files. Everybody knows how to generate comma separated value. We use those as a template, and we automate the transformation from that into that. Right, so this is our lingua franca, right? And it works. Can I prove it? Does this work in practice? So here are 5 fair data points from partners representing neuromuscular dystrophies. These 5 fair data points, one of them actually has real clinical data. The other 4 are using mock data at the moment. And then we say, okay, what question can we ask that is privacy preserving, by the way? The question was, what is the delay between symptom onset and diagnosis? Turns out this is really important for the rare disease community. The diseases are rare, so the clinical diagnosis is sometimes delayed by years as people get misdiagnosed or misunderstood. And so the delay, the offset between the first symptom and the correct diagnosis is a measure of the quality and efficiency of the healthcare system. So this is a really important metric for them. So turns out this question can be automatically answered. The metadata allowed my query agent to discover and interact with the participants that were capable of providing the question-relevant data. I then issued the same query to all of those participants because we have a shared model, and then I integrate the output, and you can see here that we're getting answers from 2 of the 5 partners who were capable of answering that question. Caveat emptor. Right. I've said it a couple of times, but now I'm going to say it explicitly. Fair data alone is not sufficient to achieve interoperability. I'm going to prove that again. This paper. So this was the European Joint Program on Rare Disease, collaborating with a group in the United States called CPATH. We did independent verification efforts over exactly the same data, and then we tried to cross-query. Well, to say, I use the words emergent here. Now, in English, we have an expression which is putting lipstick on a pig, right? Emergent is the most generous word we could use. right? There was almost no interoperability whatsoever between these two data sets, both of them beautifully verified, but independently modeled. So now we know that shared models are in fact necessary. This may be influenced a little bit by large language models who we think might be able to negotiate between these, but at the moment, we need shared models. Okay, so now I'm going to segue into the panel session that's coming up next. I want to talk to you about Flare GG. So we're now going to look at big data, networks of federated data leading to conservation strategies. So thank you to the Gobierno for funding this project. So this is verification, linking, and integrated reuse of global ex situ plant germplasm, seed banks, seeds. So our first target is the Banco de Germoplasma Vegetal in UPM, one of the most complete collections of wild crucifers in the world with more than 1,000 taxa, seeds of Iberian and the peninsula in general, currently preserving 24% of the threatened flora in Spain. Now I want to contextualize this a little bit. So we are a wild germplasm bank, and this is a model sort of circa 2009. It's somewhere in the middle of the development of Eurisco, and I'm sure there's people in the audience who will know this history better than I do. So Eurisco was hosted at the IPK Gadersleben, and this is for crop seeds, so not wild. Right? And so there was this flow from the national crop gene banks into national focal points, which then reported to Eurisco that made this general search catalog. Everything was really quite nice. The data flow was nice, very well organized, fairly well resourced all the way through here. But then we sort of circa 2022-ish, there had been a decision that it would be really nice to get crop wild relatives into this, what was formerly just crop information. Now, crop wild relatives, another English expression, we set the cat among the pigeons. This is a lot more chaotic than this. You have NGOs, you have research centers, you have citizen science, you have universities, many different data formats, very little coordination, and just for anyone who's listening, very little resourcing. They need funding. So this plethora of new stakeholders with highly varying expertise and varying levels of compliance with the emergent standards from the FAO, I'm sure is also here. So the FAO had created descriptors for crop wild relatives that were supposed to allow this movement of data. It was still difficult, and there's these other issues that came up, the legal regulations on territories, legal regulations on species, conservation status. Now, I'm not going to talk about this, but my lab is really interested in modeling this for machines also. So if you want a conversation about that, catch me in the break. So this is a mess, right? But it's identical to the situation that we face with the registries in the EJP. Exactly the same problem, including the privacy issues, by the way. And I want to just note that FAIR data facilitates communication between these partners, in addition to potentially lubricating this movement of germplasm information from the crop wild relatives germplasm banks into Eurisco. So we're helping in many different ways if we go through a verification process. So the objectives are to take what we learned from EJPRD and now apply it to germplasm banks with the BJV as our first target. And then we're going to expand. So let me show you the status of this project, and this will be the last few slides before I summarize. So we have our FAIR data point. This is what it looks like. It's customized to the germplasm case. There's an automated installer available. We have metadata capture templates available. And we are also offering to host. So if anyone wants to join us and they want low cost of entry, we will set it all up for you. And you can play and then choose to host it yourself later or not, as you wish. We have the models. So we've created the data models for all of the different kinds of germplasm data. They're shared. We have the CSV templates. We have the transformation pipeline tested, confirmed that it works. And the entire infrastructure can be replicated by any partner within a couple of minutes. There's an automated installer that does everything. We have our index, and so far, we are very lonely. There's just us. Right? But this is early days still. We've only just built this in the last few months. So there's several seed bank partners. This is coming mostly from Red Bag. Partners identified who will be on board with us in a few weeks, we hope. But in fact, everyone is welcome to participate. Contact me. By the way, you don't have to contact me at all. The instructions for getting on board are on the web. And we don't really, at the moment, have a curation process. Now, we will, of course, watch and make sure that only valid people are registering, but we're trying to not centralize anything. This is an open, fair, distributed network of partners, right? Self-selecting partners. And then the thing that we have here is what I call the virtual platform. So this is the entry point for exploration of the entire partner network, but what it does is it drives traffic outwards. So when you do discovery searches, it says, they have an answer, they have an answer, they have an answer, they have an answer. Right? And so we're driving traffic to the individual resources rather than centralizing. And that's important because they need visibility. They are really in trouble in some cases. Right? So they need visibility. And finally, this is the coolest part. One-click launch. So once I've done my exploration, I've done my query, I can click and launch an analytics platform, an analytics environment, which is customized to the question that I asked. Right? And this is possible because the services that are asking the questions are also annotated fairly. So the system knows what question it just answered, and it can automate the construction of an appropriate analytics environment, for example, map integration. So the next steps are to begin constructing shared queries that are intended to help build conservation strategies, and now we're getting into the topic of the next panel. So let's look at what that will look like. Dynamic integration of partner seed bank collections with, for example, GBIF species observation. So in this map, you'll see the yellow dots are all the locations where a particular species, this is Caricitera, have been observed. The red dots are the locations where it has been sampled in Spain. Right? So observations are not the same as samples. Right? And so from here, when we get these maps together, we can say what geographic locations have not been sampled by any collection expedition from anyone in the network. Right? So this is a breadth strategy. What have we not seen yet? Then we go deeper. Right? So we know that environment drives intraspecies diversity. So now we're going to add resources such as AMET, the Meteorological Agency, the IGME, so the Geological and Mining Institute, Spanish Geographical Agency, and capture contextual information, environmental contextual information associated to where species live. And so you can see that we can now subdivide these observation locations into observations with different microclimates, with different soil types, with different salinities, and so on. And then we can say, are there occurrence locations of a particular species of interest within which we haven't sampled? For example, a breeding program might say, well, I'm looking for wild relatives that are tolerant to saline conditions. And we could look here and say, well, here's a region that hasn't been sampled at all that matches those conditions. So the genome has likely been adapted, and we can sample from there. So this is, again, telling us what we don't know, which is very cool. Right? It's a different way of looking at big data. What don't we know, not what do we know? Okay, take-home messages. FAIR is metadata first, metadata first, metadata first. FAIR is measurable, but for the moment, don't trust it. We need global governance before you can really start to trust these quality assessments. FAIR data alone does not lead to interoperability. We need shared models. Rich, high-quality metadata enhances the appropriate, I'm going to use that word again, I'd love to explain it later, reuse of FAIR data. There are technologies and strategies that allow FAIR experts to help you, help non-FAIR experts, help anyone to create their own FAIR data, their own really well-crafted FAIR data, by the way. So that allows us to distribute efforts rather than centralize or warehouse these FAIR data. And finally, don't reinvent wheels, please. So projects like EJPRD, lots of money in EJPRD, have generated a mountain of reusable code and models that you can apply to your own verification efforts. And that's it. So I have numerous and treasured colleagues and collaborators that I've been citing throughout the slide deck. I want to especially recognize the Ministerio for funding the FLAIR-GG, the European Joint Programme on Rare Diseases, and of course my institute, CGPG INEA, CSIC, at the Universidad Polytechnic de Madrid. So thank you very much, everyone. I don't know if I'm supposed to take questions or just go straight into the panel. So maybe.
Yes.
Am I supposed to take questions or just go straight into the panel?
No, take the panel.
Okay. so it's very bright. Questions from anyone?
It's okay.
Okay. Thank you very much.
Thank you. Thank you, Mr. Wilkinson.